Good morning, good afternoon, good evening, colleagues and friends from all over the world. Welcome at the second edition of the webinar series, Advancements in Cancer Imaging. These webinars are organized by the European Society of Radiology and the European Institute for Biomedical Imaging Research. We have an interesting program for those professionals and scientists working in the field of oncology want to stay updated on the advancements of AI in cancer imaging. So we hold these webinars on a regular basis, so also look out for it in 2025 because there will be more coming. Today we will focus on data repositories for AI evolution in cancer care, and over 1,500 registrants from all around the world have already joined us. So thank you for that. I am Regina Beets-Tan. I'm the Scientific Director for Biomedical Imaging Research. And I would like to first explain you what is EIBIR. And I'm going to move forward. So EIBIR is the European Institute for Biomedical Imaging Research. We are a non-profit organization which help to bring together researchers from different disciplines and nations to collaborate in biomedical imaging research. EIBIR is founded by the European Society of Radiology, and for those who plan to conduct EU projects, we can help you navigate through the landscape of the projects and help you access funding for research. So we can take on coordination and management and dissemination of your research. So you can focus on the science. EIBIR has over the years become a real quality label for biomedical imaging research. Now, today you will learn about 3 projects that EIBIR is conducting: the CHAIMELEON project, the EuCanImage project, and the EUCAIM. The CHAIMELEON project aims to develop a structured repository of images and related clinical and molecular data on lung, breast, prostate, and colorectal cancer. This repository will contribute to the development and validation of AI tools in cancer care. The EuCanImage aims to create a large-scale, federated and secure European platform for cancer imaging, focusing on developing an interoperable platform with AI tools to support researchers and research and cancer care. And the EUCAIM is a flagship project of the Europe's Beating Cancer Plan and establishes a federated pan-European infrastructure for over 60 million cancer images, which will be accessible to clinicians, researchers, and industry for the development and benchmarking of trustworthy AI tools. And I'm pleased to have the leaders of these big projects joining me to explain you all about it. First of all, we have Professor Martí Bonmati. He's the Director of Medical Imaging Department and Chairman of the Radiology at La Fe University and Polytechnic Hospital in Valencia. He is also the project coordinator of CHAIMELEON. His lecture is focused on accelerating the AI tools from lab to market for enhanced cancer management in radiology, and he will share us the achievements of the project. And his lecture will be followed by Ana Jimenez Pastor. She's a biomedical engineer and technical manager of the CHAIMELEON project, and she will share with us a use case in this project. Then we will develop deeper into the EuCanImage project. And the focus of this presentation will be on building the future of AI-driven precision oncology with integrated cancer imaging and biological data. And this will be given by Professor Karim Lekadir, who is the director of the Artificial Intelligence Medicine Lab at the University of Barcelona, and he's the project coordinator of EuCanImage. And we are very multidisciplinary, so I'm very pleased that Professor Maciej Bobowicz from the Medical University of Gdańsk in Poland will join us. He's the work package leader in this project and a surgical oncologist working at the radiology department. And last but not least, you will learn from the EUCAIM project and especially the sustainability of the data repositories on the EUCAIM from Mario Aznar, who is a CEO at Medical Innovation in Spain. So a very, very interesting program, and I hope you will enjoy it. I'm sure you will learn a lot from our excellent speakers. So without further ado, I would like to give the floor to Professor Martí Bonmati with his first lecture. Thank you very much, Louis, and please take the floor.
Okay, can you see the slides?
Yes, very well, and we hear you.
Okay, is it on the presentation mode? Okay. So,
yeah.
Now much better.
Yes.
Yeah.
Okay, thanks a lot. Well, thanks a lot, Regina, and congratulations for EIBIR on this huge effort to link and lead also so many projects and link the projects trying to share what are we doing on data repositories and the strategy behind those projects. What I will share with you today is how CHAIMELEON is doing and what's behind the project. CHAIMELEON is aimed to accelerate the lab-to-market transition of AI tools for cancer management, which is extremely important because in most of the research projects, we stop on the research tools, but it's mandatory to innovate and go to market to allow all the hospitals and clinicians to use the solutions we did provide on the research environment. To do that, we constructed a consortium of 18 partners from 9 countries. The coordinator is my hospital. We will finalize February next year. Total funded was 8 million. And the complexity behind that is how to link clinical centers, universities or institutes and companies, enterprises, both large and small, with the aim to populate a large centralized repository to be able to provide with harmonized medical images to avoid the reproducibility crisis, allowing the universal use of the software, and also how to proceed to the regulatory issues, how to, at the end, have a product that is able to be used clinically. And most of the centers are high-level reference hospitals as the institutions and the companies. And this is the project overview. We did create a cloud-based repository of cancer images. As you will see later, the CHAIMELEON repository is linked to the EUCAIM infrastructure. And we create this repository to allow AI developers to work on the developments of solutions that have to be used in clinical practice. And how does it add to the current state of the repositories? Before we go into this high quality, highly structured clinical data linked to images, the 5 types of cancers we are working on are breast, lung, prostate, colon, and rectum tumors. And by working on this, we were discussing all together on the interoperability of the data. I mean, how the repository we are constructed can be linked to other imaging repositories or biobanks. We were also working deeply on the computational power, meaning how to incorporate all the functionalities within the AI experimentation to be able to provide solutions without the need to download data. I mean, being on an extremely secure processing environment. And we were also working on how to incorporate cutting-edge functionalities and tools allowing this multi-centric, multi-institutional data set harmonization, enhancement and integrity of interpretability of the solutions, meaning that we are working deeply on explainable AI and preserve privacy. So the project implementation phases were divided or split into designing and setting up the infrastructure of the repository, populating the repository with data, which started since the beginning and still is open. We are still today, a few months to the close of the project, still requiring external providers and internal providers to populate the repository. We are implementing AI-powered solutions and functionalities within the repository, performing a clinical validation of selected solutions, having legal support within GDPR compliance, and creating also a long-term sustainability business plan, as we will see later. This is an example on prostate, but we have the same examples for breast, lung, colon, and rectum of the patient's journey, the pathway patients face in clinical practice, which means from diagnosis, local staging and distant, first treatment evaluation and follow-up, and the type of variables of information that we were gathering from the electronic hospital records, the data warehouses, to be able to solve a relevant clinical question. And for each type of tumors, clinical objectives were defined. For prostate, it was precise tumor staging and grading for treatment allocation and prognosis at first diagnosis. Then we define inclusion criteria, exclusion criteria, and an expected number of cases. The repository population strategy was split into phases. The first phase was internal partners. We were able to provide, or we were willing to provide, or asked to provide at least 13,000 cases of high quality with imaging, clinical data, and clinical endpoints. And the mission was to populate the repository, allow AI developers to develop the decision support system tools and assist on those clinical questions. And we need external cases, mainly with imaging and minimal annotation, to even help the developers on those aspects that do not need clinical correlation, mainly related to image harmonization, image segmentation, and image annotations. These are the external data providers we did incorporate within the project, providing a larger amount of data, and most important, from many more institutions, and be more representative of the real world. So these are the complete cases and the additional external cases provided. Our objective was 13,000, and already we do have 60% of the cases already in the central platform. And our objectives, I'm pretty sure we will close it, will be 21,000 patients. So this is the phase of validation. We did have an open challenge. We exposed the repository to researchers outside the project to develop clinical decision support system on risk stratification, histology subtypes, TNM categories, invasion versus non-invasion on rectum, and overall survival. And this is the background on how many participants we did have, coming from so many different institutions and countries to develop the clinical decision support systems. And those solutions, developments on AI tools were also pushed by in-house developments, mainly at Maastricht and my own university, also to compare with the winners of the Open Challenge. And our aim was to ensemble deep learning and handcrafted radiomics, explainability of AI solutions, comparison of harmonization approaches, and foundation models. Having those solutions after the Open Challenge and internal developments, we did perform clinical validation of the models, and we asked radiologists and clinicians and oncologists to check how were they using the clinical decision support systems and the clinical gain in the decisions when those systems were used. And we did compare timing per case, results biases, and collect feedback and comments. And we are so happy we did develop this assessment of AI trust. How much did clinicians and radiologists trust on the solutions, again, for prostate, lung, breast, colon, and rectum. And the main resource for that is that at least with the solutions we do have and the data we expose to the for validation, the solutions are highly appreciated, which was good. And I don't want to finalize without sharing with you one important aspect of the CHAIMELEON project, which was a re-identification challenge. We are fighting all over the research projects we are engaged with the issue of GDPR and the risk of re-identification. Although if you go back to the publications, there is really not so many publications on which is the feasibility to re-identify a patient once part of the clinical history or images are exposed to the community. In some way, we did recognize that the regulations do not provide technical information on how DICOM images should be really anonymized. Some organizations propose different methods. Within our own research projects, we make home adaptations of those solutions, and this de-identification is a hot topic today. And we were really willing to understand in reality, which is the risk exposing data to the research community. So we introduced this challenge. We opened the challenge to researchers within AI for Health Imaging initiative and the EUCAIM infrastructure project to researchers, asking them to try to re-identify anonymized DICOM studies. And we used two de-identification or anonymization methods, one based on CHAIMELEON and the other one on ProCancerAI. We were willing to validate the robustness of the anonymization process and also to assess the level of data security and usefulness and the likelihood of re-identification. And we did that on a single phase, coordinated by my hospital, also trying to keep everything under control and allowing researchers to work on the data to identify patients. And after so many months, no patient was identified. So we are extremely happy now. We are preparing a publication on the real risk of re-identification, which at least with today's level of knowledge on computational and AI issues is not really feasible. So in conclusion, for the CHAIMELEON project, we were able to construct a high quality, highly structured, with those so many clinical, pathological, genetic, treatment, and follow-up variables, data linked to DICOM images. We did develop an interoperable aspect, linking our imaging repositories with other ones, mainly with the EUCAIM functionalities and interoperability. We were able to provide computational power, incorporating all those functionalities within AI experimentation on the cloud, without the need of downloading the data, which is extremely important for us because we were willing to work on a secure processing environment. We did incorporate cutting-edge functionalities and tools like data harmonization, as we will see later with the lecture of Ana Jimenez, but also integrity, interoperability, and explainability of AI. And we were integrating the CHAIMELEON, or we are integrating the CHAIMELEON platform in the EUCAIM project, as will be shown later with Mario Aznar. And that was my presentation. Thanks a lot. I'm so happy to go back to the questions. Thank you.
Regina, sorry, you're muted.
Sorry. Thank you very much, Luis. Huge achievements, and thank you so much for sharing that. I think we will keep the questions to the after the lecture of Anna. Meanwhile, I would like to encourage the audience to just drop your questions on the Slido, and we will try to address them. So Anna, please take the floor.
Thanks a lot. Let me share the screen. Can you tell me if it's seen properly?
It's fine, thank you.
Perfect, thank you. So thank you all for assisting this webinar. After the overview we have seen from Professor Martí Bonmati regarding the CHAIMELEON project, we are going to go in depth to a specific use case. So for that, as you know, when we are facing AI-based, radiomics-based solutions for cancer imaging, we usually follow this pipeline. Now, as Luis has mentioned in detail, we put a lot of effort in data collection, so acquiring data from multiple institutions in order to have a huge variability of what we can find in the real world, which is followed by different steps. Now, going through data harmonization, which is very important, as we will see, to standardize the quality and the interpretation of the medical images, followed by organ or lesion segmentation or detection, to finally create endpoint prediction algorithm. So in this session, we are going to focus on some of the points that we have put a lot of effort in the CHAIMELEON project. So starting with data harmonization. So before going in detail to what we have done in CHAIMELEON, I wanted to do a summary or an explanation for all of us to understand why do we need harmonization when we are dealing with multicentric data. No? So when we are dealing with real-world data, we find a huge variability on the scanners that have been used, on the acquisition protocols that were followed in order to acquire the images. So we see a huge heterogeneity in the image quality. So this can introduce biases when we are extracting quantitative information, such as radiomics features, imaging biomarkers, which can reduce the AI model's generalization capabilities. So as you can see here in the image, if we have images from different scanners with different properties, acquisition protocols, we see, especially in MRI, it's also seen in other modalities, but this is very punctuated in MRI, in the sense that we see a huge difference in terms of intensities. So our ambition is going from a huge variability in terms of intensities in our data set to a more standard way of visualizing and interpreting the images. So our objective was to guarantee the consistency of any image that we are using in our AI models in order to strengthen the progress in research, by reducing the biases and the variability found in those data sets. This, apart from enhancing the research that we can do with this data set, we have also seen that this reinforces the cost effectiveness and robustness in clinical studies, especially in phase 3 clinical trials, where multicentric data are used to compare different groups of a drug, for example. And then this is the foundation in order to develop robust AI-driven predictive models. So we want to go from a data set in which we can see a huge difference in terms of, for example, intensities, but not only intensities, also levels of noise, field of view, resolution, etc., to a more homogeneous data set where all these differences are minimized. So in CHAIMELEON, we have developed different approaches for image harmonization, following two different approaches. On one hand, we have developed self-supervised learning approaches, and on the other hand, we have developed GAN-based approaches. In this way, we faced two different ways of handling harmonization problem in order to compare which solution was the best on each scenario. And in this way, we have developed harmonization solutions for different clinical indications and modalities. So let me focus on the first approach, which is the one that we follow through self-supervised learning. In this case, I'm going to focus on prostate MRI, where we can see this huge difference when, for example, dealing with the T2-weighted MRI. In order to minimize and to harmonize the T2-weighted MRI, we followed an approximation in which we, first of all, created, following a self-supervised learning approach. This means using the information of the images themselves, we synthetically created images with different intensities and resolutions. For that, we took advantage of the frequency domain that allow us to create from a reference image other alterations, more hypointense images, hyperintense images, so different alterations in order to simulate what we can find on different scanners acquisition protocols. After that, we evaluated the performance of the harmonization solution when training an AI-based algorithm for prostate segmentation. So we train an AI-based algorithm in order to segment the prostate gland on 3 different regions: the central transitional zone, the peripheral zone, and the seminal vesicles. And we conducted 2 different tests. The first test was based on T2-weighted images, excluding images with endorectal coil. So here you can see what the performance in terms of desk coefficient when we train the segmentation without harmonization and with harmonization. In all the different areas, we found an improvement in the performance in segmentation when we used the harmonization approach, and the differences were statistically significant. However, when we went to a very different scenario, what we consider like an outlier, this is the case in which we only used cases with endorectal coil, as you can see here in the test tube, this increase in the performance is much more significant. This is something that we can also see here in the images. If we use an AI algorithm trained with images without endorectal coil, we can see that the generalization capabilities of our segmentation solution is quite bad. However, after conducting harmonization, this is the images that we can see, and we can see as well that the generalization of the algorithm is much high. So in this way, in the project, we conducted harmonization solutions both for CT, especially harmonizing the resolution, and for MRI, especially breast MRI, rectum MRI, and prostate MRI. And following up in the pipeline, the next step in which we have put a lot of efforts is as well in the endpoints prediction. As Luis has mentioned, we conducted an open challenge in which external researchers trained AI models in order to answer different clinical questions. We had different diagnostic clinical questions for prostate, breast, colon, and rectum. And also we had a progression clinical question in which for lung cancer, we aim to predict the overall survival. Let me focus on one of the solutions that we have created for prostate, in this case using prostate MRI to predict risk of a patient. This is a work that was led by the University of Maastricht, in which having the T2-weighted image, we performed a segmentation of the prostate gland, and then we used an ensemble, a combination of different models in which we use images with different resolutions, and as well, we took advantage of the power that the foundational models can give us in order to build a predictive model to predict the risk of prostate cancer patients. And here you can see the performance that we have today. If we compare the different models against the ensemble, we can see a huge improvement in the performance. We're combining all the models together. So we have today an area under the curve on 72%, which we continue improving once we have more data in the repository. So this is the summary now I wanted to show you regarding a relevant use case we are conducting in CHAIMELEON, putting a lot of efforts in the harmonization of the images in order to guarantee that we can extract the highest power as possible of our data set. So thank you for your attention and open to any questions.
Thank you very much, Anna. Let's open this 10 minutes for questions from the audience. So I already have one that I would like to address to Louise. Are these data, and there are several questions on this, are these data of the project publicly available, and how can it be scalable to other EU research projects? And I think we are referring also to the EUCAIM. Can you elaborate on that?
Oh, yeah, pretty sure. Thanks, Regina. And thanks for that question, because that's, let's say, critical at least European level. I mean, every time we are conducting a research project, after these 4 or 5 years, all the efforts, the data and the tools mainly disappear. I mean, they go nowhere, and we are not able to reuse that data for further research on data. And we did have within EUCAIM, as within CHAIMELEON, as on all the other projects, work packages on sustainability, how to expose data to other researchers. But the reality is that it was not done. And to avoid that, we started the EUCAIM project that we will discuss later at the end. And we are deeply working and already agree within CHAIMELEON that the data and the tools, those tools that are available and produced during the project with the licenses and whatever related to the partners that generated those tools, will be transferred to the EUCAIM infrastructure or platform to allow researchers to work on that. And the main reason is because there is no more money to keep that data available, and those tools also available within the research project. The research project is finalized and money and data disappear. So to avoid that, we are really extremely proud that the consortium within CHAIMELEON agree on data sharing now that we are active, but data transfer later once the project finalized. And not only data, but also tools to the European Cancer Imaging Infrastructure Consortium.
Well, very clear, Louis. Thank you so much. Just one question to Anna. The participants ask whether there are also plans in this direction for brain tumors.
In the case of CHAIMELEON, as we have commented, we have focused on prostate, lung, breast, and colorectal cancer. However, the same methodology can be applied to other areas, anatomical areas, modalities in terms of MRI, or areas on CT scans. We internally in the group, we have tested how this same methodology is applicable for prostate MRI, rectum MRI, breast MRI. So the same methodology was followed. So exactly the same methodology for sure can be used for brain tumors as well. So absolutely, the same methodology that we have followed is generalizable to other anatomical areas as well.
That's very good news. Luis, a last question to you. There is one participant colleague from Turkey who would like to understand or to offer their center to join this project. In what kind of role could that be? And maybe this is also a question from other centers that are now joining us. Can you explain a bit on this procedure?
Yeah, pretty sure. Well, we have to recognize that research projects are funded by the mainly European Commission, these big ones. So partners receive money to do tasks and make developments. And being part of a research project once it's started is extremely difficult or impossible. Even notice that we did open CHAIMELEON to external hospitals and data holders to allow them to be engaged within the research community. And we are so happy and proud that 10 hospitals were already engaged within the project. One of them from Istanbul, the Istanbul Medipol University Hospital. But CHAIMELEON is close in some way. We are going to finalize now. What is extremely important is that even now we understood that so many hospitals like these ones you are telling, no, Regina, are really willing to participate in the research community. And the way to do that, even if we are not deeply engaged on a specific research project, is to go through the EUCAIM webpage to ask for having being a stakeholder, being a data user, a data holder, a data provider, a tools or AI environment, because we do have those pathways on EUCAIM as an infrastructure. So if you are not on a research project for now, you are always able to be engaged using the EUCAIM platform.
Thank you, Louis. It's so great to hear that a project doesn't close and end, but there will be another project which is even larger, building on this project CHAIMELEON. So both Louis and Anna, congratulations on the achievements, and thank you very much for your input in this webinar. Now, I'm very happy that we can delve deeper into the EuCanImage project, and Karim will talk you through the building of the future of AI precision oncology with integration of imaging and biological data. Please, Karim, take the floor.
Yes, good morning, everyone. Thank you again for the invitation. It's a pleasure to be here to tell you about the EuCanImage project. So this is one of the 4 projects of the AI for Health Imaging program, together with CHAIMELEON and Incisive and ProCancerEye. In this project, we have 20 consortium partners. We have the research infrastructures, where the data is stored and managed, Eurobioimaging, European Genome Phenome Archive, BBM Archive, and the American infrastructure, the Cancer Imaging Archive. So we build on existing archive instead of developing something completely new. We have academic institution, University of Barcelona is the coordinator. We have also industry partners, including a big company, Siemens, but also small companies. We have 5 clinical sites from Spain, Poland, Lithuania, Sweden, and Italy, so cover different regions of Europe. And of course, we have an important European organization to help us in particular with the dissemination, communication, outreach, and networking, including EIBIR. So the first goal of the project is to build a platform that can link imaging data with non-imaging data. Now, the figure looks a little bit complicated, but let me explain it to you. It's in fact rather simple. So on this side, we have the clinical sites who contribute data to the platform. Here in the center, this is where we store the data. So at the EuroBioimaging, the XNAP, the imaging data go there. In European Genome Phenome Archive, the non-imaging data go there. And the metadata, that's the catalog, is stored at the EIBIR imaging catalog. So the data sets are pushed from the clinical sites. First, they are anonymized and curated using the well-known PostDA suite and annotated using the collective Minds Radiology platform. Then they are managed centrally in these existing infrastructures. Later on, this is the second part of the project, the AI developers come in, apply to access the data set, so that could be also third parties, such as some of the participants in this event. This could be AI developers that would like to build new AI tools, but also validate them. And OpenEbench is a validation environment. So to get there, we had to follow a number of steps. The first one is we set up a contractual framework. Now, this is a challenging step, but a very important one. Of course, we have to comply with the GDPR, for example, but also with national and local regulations. So this led to a contractual framework starting with the clinical sites, that the data providers that provide the data, they have a contract with the infrastructures, the European infrastructures, EuroBioimaging, European Genome Phenome Archive, where the data is stored. They have also a contract with the data platform where the annotation is done. And then finally, with the end users, those that will request the data, this could be partners of the consortium, but this could be third parties here. So this could be anybody that's not in the consortium that could apply to use the data, and then would access the data through these contracts that are now readily available. To do all this, we built a number of clinical use cases, 8 clinical use cases in 3 domains: liver cancer, colorectal cancer, and breast cancer. So this has allowed us to build a data platform that is flexible enough so it can handle a diversity of imaging modalities and clinical use cases, but also focused enough so we can develop something that is actionable after the project. So the project will finish in September of next year, and the goal is to reach 25,000 new data sets classified across these 3 cancer types, and also to have them all annotated. And we are almost there. We have all the data identified and anonymized and curated. Most of the data is annotated, and we are now finalizing the process so we can provide high quality, fully annotated data set to the communities that they could use in the future to build new AI tools, but also to validate their existing AI tools. This is how the platform looks like. It's a web portal. It has different services. So, for example, it has a catalog where you can search for the data. It has an access request manager where third parties can request access to the data. There is the annotation platform that I described of Collective Minds Radiology. There is the AI virtual research environment where new AI tools can be built. But there's also a benchmarking environment where AI tools performance can be compared by using reference data sets. This is an example of a data set that is described in the catalog. So it has a lot of information, and users can then decide whether this is a data set that is relevant for their research or for their AI tool development, or they can search for other data sets, and then they can also request access to the data set. This is the annotation platform that we used. It's a collaborative platform. Clinicians and radiologists can connect from different places. It's fully GDPR compliant, very much user-friendly to facilitate this process of data annotations. We also put emphasis on quality control, so we want to ensure that the data set is high quality. So we built on existing tools, in particular our collaborators from the TTIA, the Cancer Imaging Archive, who are part of the project and who have unparalleled experience in this field. They built the Cancer Imaging Archive for more than a decade in the US. So we built on that experience, and we built these tools to ensure that our data set is of acceptable quality, so it can be used efficiently in subsequent studies. I mentioned already the virtual research environment. This is where we build and provide AI tools that we develop. So Maciej later will give you more details on the clinical use cases, and you will see what sort of AI models we are targeting. This is just as an illustration, example of results we obtained. This is a study to obtain generalizable model. So we have 6 data sets in breast mammography. Therefore, there is a problem of generalizability. These are different domains. There is quite important heterogeneity across these data sets. And we build new techniques that allow these AI models to be used across these different domains. So before, there were errors in the detection of the tumors, and they were corrected by using domain generalization techniques, for example, transformers or transfer learning. So I mentioned one target of our project in terms of AI, which is generalizability. Other dimensions of trustworthy AI that we've worked on. So for us, the performance of the AI tool is important, but trustworthiness and ethical compliance is very important. That's why we built this framework, this guideline that we call the Future AI Guideline, to make sure the tools are fair, universal, traceable, usable, robust, and explainable. We did this within EuCanImage, but also in collaboration with the other project of the AI for health imaging, including CHAIMELEON and the other projects, but also EUCAIM. And we also invited collaborators from all over the world. So this is very much an international effort. If you go on the website, futureai.eu, there is a link to the manuscripts, to the preprint, where you will see that we give a step-by-step guidance on how to ensure the AI tools are not only accurate, but also adhere to these principles of trustworthy AI. And finally, once we have the AI tools, we would like to validate them. And in particular, we'd like to do benchmarking. Benchmarking is a very important activity in scientific research. It allows to compare objectively different AI tools and AI models by using reference data sets, and we build an environment, open eBench, that allows us to do that, and which was included as one component of the project. So this is the end of my presentation. Maciej will tell you a bit more, we'll go into more of the details of the clinical use cases, and I'd be happy to answer your questions later regarding the platform and the technical developments of the AI tools. Thank you for your attention.
Thank you very much, Karim. Very nice achievements of the EuCanImage project. So let's go to Maciej, who will explain a concrete example of a relevant use case. And meanwhile, please, audience, drop your questions on the Slido. All speakers will stay and remain here until the end of this webinar. Thank you.
Thank you very much. So you already heard a lot about our project, EuCanImage. It's a wonderful project. As Karim mentioned, we are concentrating mainly on 3 organs and diseases related to breast, colorectal disease, and liver disease. as we are mainly focusing on cancer, we collect data on cancer patients, but also have cohorts for patients with benign disease and also completely normal studies. Because as you might be already aware, if you are trying to develop some AI algorithms, they have to have something for comparisons, right? They don't work just on detection of something that's there in every single image. So we have to train it properly. So for me, the most interesting part of it is breast cancer. That's the biggest data set we collected. We have altogether over 8,500 subjects, so patients in the platform for breast cancer. We also received additional data set from one more hospital from Spain, which contains almost 3,000 more patients. And I will refer to it at my last slide in the presentation. But what we are doing with these patients, because we concentrated 5 years ago when we wrote the project on mainly detection of the cancer, right? And the proper classification of lesions in oncological imaging. So in here, we concentrate on mammography. You can see that we are collecting CC and MLO full field digital mammography images for both breasts for each patient. So we have 4 images from each patient, which makes over 40,000 images already. To make the data reusable for different other purposes, we decided to go for pixel-wise segmentations. And as Karim already mentioned, we use this Collective Minds radiology platform for distributed segmentation and annotation of images that allows you with some additional support to do those pixel-wise segmentations. We segment in mammographies almost everything that's there. So multiple pathologies. We segment cancer, benign lesions, microcalcifications, foreign bodies as implants, tissue markers, but to be true, everything that's in images. That was a very immense work from the whole radiological team working in EuCanImage that I have to congratulate and say a huge thank you to everybody who participated so far. The radiologists also supported us with radiologist reports, not only images. So they have BIRATS charts where they also supported it with the reviews. They supplemented ACR's density for every image. And they also had free text spaces where they added remarks that we didn't think about in the beginning of the project. And as Karim mentioned, we collected also a lot of clinical variables. For this use case, we collected 35 different variables, and we had 2 different objectives for this. One is our project, where we had 8 different clinical questions, but we also wanted to make it reusable and, to add some extra value for future generations and future research. As you can see in here, we use different tools. Some of them were a bit more automatic, using ETLs in some of the hospitals. Some of the hospitals supplemented data with manual forms done as ECRF forms in REDCap tool. All this data is stored in the European Genome Phenome Archive for future artificial intelligence modeling and secondary use. All the data, as already mentioned in previous presentations, has to be useful, right? So we have not only minimal sets with ground truths, we have additional information that is really necessary for our use cases, but we also have some additional information, additional variables for the future projects that can be done on this huge data set. And as Karim already mentioned, we managed to implement the data quality module for both clinical data and imaging data to make sure that the data is robust and it's complete, right? It doesn't contain any mistakes. When talking about images, we also have to check the imaging quality. As radiologists, you are very well aware that not every single imaging study is good enough for diagnosis. So sometimes you have to repeat it or reject the study you are just reviewing, right? So the same in here. So we asked radiologists to perform a Likert scale assessment. They assessed if the study is of a diagnostic quality, but that's not enough. Apparently, AI algorithms can't work with it sometimes. So even if you can assess the study, you can say there is cancer, you can tell the staging, do the CT&M staging. AI algorithms sometimes don't work on those studies because they contain some kind of artifacts. That's why we used also automated quality assessment with image acquisition protocols and peak metric scale. And there is one paper coming soon, hopefully, where you will see the results. And apparently, those metrics not always overlap. So something that is usable for radiologists might not be good for AI algorithms development. And we'll explore it more, and you will be able to read it, hopefully, in the near future. So that's the breast mammography data set. It doesn't include the 3,000 patients from this additional center that I told you already. And as you can see, there is almost 8,500 subjects with more than 10,000 images per site, so over 40,000 single images. It's supported with structured report formats. It has segmentations. It contains a lot of clinical variables for every single patient. And in here, you have a small snapshot for the data quality checks for my center, GUMET, and Hospital Clinic Barcelona. And as you can see, we checked for breast density in here. And as you can see, the distribution of breast densities is slightly different. That's why it's great that we got data from so many different European countries, because it varies, sorry. And as you can see, we have at this stage, we are reviewing the data. So for example, in the right part of the slide, you can see U, which is undefined. So we are trying to recover this information from health systems to provide as complete a data set as possible. And as always, in large projects like ours, we have challenges and limitations because we included a lot of screening population as a huge part of our data set. We know that some populations might be slightly underrepresented. So a lot of our patients are at the screening age of 45 to 70 years, and outside there will be slightly less patients in the data set. And then when you request such a data set, access to the data set, you will have to think about such challenges and limitations. On the right-hand side, you can see a lot of challenges that our radiologist team encountered, such as foreign bodies, different abnormalities that are not really related to the breast itself, and caused a lot of trouble when segmenting the images, providing additional work for people. And as Karim already mentioned, we supplied this metadata catalog to make the data searchable. And as you can see, that's really pretty extensive amount of clinical variables. We wanted to make it as fair as possible. So we provided a lot of different meta tags for each patient to allow different AI development and a different research in AI and also clinical research on the data sets we collected. You could see this slide already. It's a virtual research environment that was developed inside the project. As I mentioned in the beginning of my presentation, we concentrated mainly on the mass detection, mass segmentation, classification, so malignancy prediction, but we also included some modules for treatment estimation. In here, we have one additional use case that I show so far, where we explore possibilities of AI being able to predict treatment response to neoadjuvant chemotherapy in breast cancer patients based on pre-treatment contrast-enhanced magnetic resonance. We also provided a toolbox for machine learning and a module for radiomics explanations that will be available to the researchers worldwide. This VRE is quite interesting because it works not only on our data, but it's connected to the Cancer Imaging Archive, one of the partners of our projects from the United States, and you can also upload your own data sets. So it's very handy to use. And that's just a brief showcase of one of the papers from the beginning of the project, where we didn't have segmentations and explored some weakly supervised approaches. And as you can see, the results are encouraging. We are still working on it in different teams. So hopefully we'll have some better results to show pretty soon. And that's the last thing I mentioned in the beginning. So we have these additional data sets from some centers that we are requesting or are seeking for help from radiologists from Europe. And we are running this project on collective annotation. So if you are a breast radiologist, liver or colorectal radiologist, and you would like to participate in some research, please contact our colleagues from Pisa University. You have emails in here. We are asking to segment just 100 mammograms or 30 liver CTs, up to 3 lesions, and we can offer you a co-authorship of one paper when the project is successful. So if you are interested, please get in touch with us, and we'll try to work together. Thank you very much for the time.
Thank you, Maciej. That's a very good call for the audience. So while you are a surgical oncologist, so you're really treating the patients, can you tell us, because we invest a lot and radiologists would like to really contribute to this all these projects, can you tell us, well, how important is it for you as a clinician to make your treatment decision making after beyond the research period?
So we have those 2 use cases on the treatment response prediction. I must say they are kind of like high risk use cases, because that's something that is not frequently published, and it's a high gain, right? So if we manage to develop tools that we didn't have before, like treatment response, that will be just a perfect solution for us as clinicians and our patients, to be true. So that's the biggest issue. For clinicians, tools for automatic detection are also perfect when you are planning your surgery, and your radiologists are not always available at the time of the surgery. You can use some automatic tools just to tell you where the lesion is, to make better planning for your surgery and treatment. So all these tools developed in our project, but also other AI for health imaging projects are very, in our opinion, will be very useful when the tools are there.
Okay, that's good to hear, because it doesn't only stay in the research, but will go towards the area that clinical or physicians will use these tools for the benefit of patients. There is a question for Karim. Karim, how can EuCanImage be used outside Europe? How can the clinicians or researchers from outside Europe contribute to the project?
Yes, we are very much open to collaborating with research outside Europe. So we already collaborate with the Cancer Imaging Archive. So it's a part of our goals. I think the easiest way is to contact us to understand how you would like to contribute. Would you like to be perhaps contributing data to the platform? That's one option. The other option would be that you would use the data that we have. In this case, you need to apply to use the data. If you allow me, I would also reply to the other question at the same time, because it's very much related. I think there was a question in the chat whether the data is already available, whether the portal is already available. So if you go to the website, there's a link to the portal. So the portal is there, but it's still in testing mode, so to say. We are testing it internally, but we already published it externally, so people can access it. And if you want to give us feedback already, that would be much appreciated. The project is still running until September. By then, we would have a final version, hopefully, that we will then publish for everybody, hopefully a more advanced version. At that stage, all the data will be available. There will be a mechanism to request access. So if you're a researcher within Europe or outside Europe, you could request the access. Then whether you can access them if you are from Europe will depend on the different clinical sites, whether their local regulations allow it or not, or whether we could find ways to collaborate. Perhaps we could use federated learning to process the data in a privacy-preserving manner. So federated learning is part of the project. So both my team, but also Maciej's team is working on this. Yeah, just I wanted to answer all these questions at once. The objective of the project is to build its platform and then to do AI with it, to do the AI with the data that will be there. This is really the two big objectives of the project, and do not hesitate to contact us to collaborate.
Perfect message. And then The final question for either of you, do you also include nuclear medicine data, so like PET-CT data, and if so, which radio tracers? Is it the routine one?
No. We would love to, but we didn't include the PET-CTs or nuclear medicine so far.
So a wonderful call also for the ones working in that field. Well, thank you so much. I would like to move on to the next part. So we have more time for discussion after the last lecture, which is on the European Cancer Imaging Initiative, will be given by Mario, and he will share the sustainability of this project. Please, Mario, take the floor.
Thank you very much, Regina. It's a pleasure to be here with all of you today and to be able to introduce you to the European Cancer Imaging infrastructure. The goal of this session, of these 10 minutes, is to provide you with a big picture of what this infrastructure is, the why, the now, the current situation, and what we envision for the future. So the formal definition of EUCAIM is the following. EUCAIM is the European Federated Cancer Imaging Infrastructure. It is a flagship action of the European Cancer Imaging Initiative, part of Europe's Beating Cancer Plan. Okay, but why? Why Europe is investing in deploying and sustaining this European Federated Cancer Imaging Infrastructure? This is the important question. Okay, as we all know, there are millions of medical images populated in the European Union in this case, but we can use this for the entire world. Every year, there are millions of medical images populated every year all over the planet, from clinical practice, studies, research projects, cancer screening programs, etc. However, they are distributed into different hospital repositories, etc., resulting into scattered, heterogeneous, and non-standardized data. This is a barrier because we need data in quantity and quality, for example, for training and validating AI. AI could revolutionize cancer diagnosis and treatment as long as we have this data available. In addition to that, running cancer screening programs or observational studies implies intense resources in terms of time, effort, and coordination. And we all know that in many cases, national, European, international R&D and innovation projects could have a great and lasting impact once finished if their scientific community and results could could be sustained. So EUCAIM aims at providing an answer to this, a solution to this. We define EUCAIM as a pan-European federated infrastructure aimed at linking and making research cancer imaging data from national hospital networks and research repositories accessible, ready, and usable. And once we have this data available, making it ready, accessible, and usable, EUCAIM will act as an accelerator for research and also for the industry, in innovation, especially focused on fostering and progressing on AI-based cancer management tools towards personalized medicine. Basically, what EUCAIM offers is the following. You can see on the left of the screen what we call the Atlas of Cancer Images. This is EUCAIM will offer access to existing cancer image collections for secondary use. This data comes from different already funded EU projects, from different repositories. This is existing data that has been harmonized, enriched, and make it ready, accessible, and usable for data users to ask for access. And then at the right part of the screen, you can see the other dimension, observational studies and cancer screening. EUCAIM could act as a facilitator for running these programs of observational studies and cancer screening, making new data available to address specific research questions, for example. And as a result of the latter, we can offer services based on data and knowledge generated. These are, for example, act as a medical devices validator, provide statistics, studies, training, spaces for forums for research communities to share knowledge, etc. So here we can see the three main target groups we have in EUCAIM: data holders, data users, and tool providers. For data holders, EUCAIM aims at offering safety and compliance with technical, ethical, and legal requirements and facilitate long-term sustainability for data and research communities. For data users, EUCAIM offers accessible quality data, access to new knowledge, and different services. And for tools providers, EUCAIM can offer visibility, business opportunities, data, and AI tools for the industry ecosystem development. Okay, let's talk now about the context, because all that we have seen is the complete product is how EUCAIM will be in the future. Right now, EUCAIM is being deployed. It's a pilot that is being deployed, started in 2023, and it will end up at the end of 2026. It is coordinated by EIBIR. The scientific director is Professor Martí Bonmati, who started this webinar. It's a huge consortium. It's 76 partners plus another 14 partners that have been added recently in an open call. So we are close to 100 partners. And the investment is 35 million euros. There are different profiles involved, from clinical sites to data providers, AI tool providers, technical oncologists, imaging specialists, radiologists, etc. So after the pilot demonstrator, at the end of this pilot, there will be some key results to be taken over for the legal entity, for the future infrastructure once it is deployed. For example, we aim at having more than 2,000 monthly visits to the catalog in the platform, to provide to be integrated more than 40 tools, and this is a very important figure, to have more than 100,000 cases available in the collection of the platform. So what happens after the pilot demonstration? After this pilot demonstrator, there will come a phase for consolidation and expansion. At the end of the pilot demonstrator, the consortium will no longer exist as such, and a new legal entity is to be constituted. And it will be in the form of an European Digital Infrastructure Consortium, an EDIC. So within this context, the goal of the sustainability area in the project is to maintain the operation of the central hub and the federated network beyond the pilot project. This is in the expansion and consolidation stage. We need to talk then about the operational, financial, and legal access of the initiative. Regarding the operational access, as any institution, organization, EUCAIM need structure, need technology, and also human resources. We need to think about the cost and management of human resources, of operating, maintaining, and updating the infrastructure, operating, training, upgrading technical teams, etc., etc. So all these costs are to be covered. Right now, I cover half of them by grants by the European Commission and their own consortium members, but in the future, these costs will need to be covered by other means. And these means are the following. We will rely on contributions from member states, and we will rely also on a freemium model. There will be functionalities that will be free for users, and other functionalities, advanced functionalities, capabilities that will be under a pay-as-you-go or payroll model. And then also, as a legal entity, EUCAIM will be able to become partner in consortiums, and it will be potentially receiving grants for the functions in those projects. Okay, now about that future legal entity. As I said, EUCAIM will be constituted into an European Digital Infrastructure Consortium. This is an international organization and European legal entity that needs to be constituted by member states, a minimum of 3 member states. We have already, in the working group that is working in the constitution of this EDI, we have already 11 participants, and more member states, more participants will be added in 2025. Here you can see the roadmap for the constitution of the EDIC. Right now, we are in the process of sending to the European Commission a pre-notification. This is a summary of the future statutes of the institution, and next year we will send the statutes to the European Commission, and in a matter of 6-9 months we will receive the response, and the most likely is that by the end of 2026 or by the beginning of 2027 the legal entity in form of an EDIC will be constituted. The central hub, the headquarters, will be based in Valencia. Here you can see some of the main benefits provided by the EDIC, in this case to the member states. EUCAIM help to be efficient, to save cost and comply by integrating and connecting this infrastructure, the EDIC, to the national health data spaces in cancer, can enable member states to accelerate the European health data space regulation in its country, and also the initiative Health Data at EU in the most cost-effective and impactful way. As I said, we can also help in running cancer screening programs, and also provide more funding opportunities for national hospital repositories and access to early information for training, grants, opportunity, etc. In addition, EUCAIM, as an innovator, accelerator actor, provides means towards a data-driven economy and support AI-based developers, innovators in the goals. And lastly, and the most important, to contribute to fight cancer. This is, in the end, the most important. And EUCAIM is an infrastructure that partners team up with you, with the community of health researchers, health imaging researchers, radiologists, associations, etc. And there are lots of spaces where synergies can be created and work together. For example, in workshops like this, in trainings, in engaging the industry, in engaging researchers, in promoting standards, good practices, policy recommendations, etc., etc., etc. You can access already to a preview of the platform in CancerImage.eu. Is already available, a preview of the platform to play with. Very welcome to EUCAIM. All of you are welcome. Join us, contact us, spread the word, and thank you very much.
Thank you, Mario. It's quite an achievement, an ambitious project, but I can see, and I'm happy that sustainability plans are already in place. I just want to ask you, we as radiologists, medical professionals, how can we contribute in having this or maintaining this sustainability system? Because you're talking with member states, but what can the medical professional do?
There are some potential answers to that. The first one is, if you are a radiologist or researcher involved in projects in cancer that are working or are creating repositories, are creating data, contact us. You are very welcome to become data holder, a data provider. And by connecting with EUCAIM, your data will be sustained in the long term, and you can continue to access to it in the future, and also keep the research community that you are part of right now. You can maintain it in the future. In addition to that, we have stakeholders boards and forums that you can contact us, and you can subscribe to newsletter and to all the updates that EUCAIM is creating in every moment. And that's it. I think you just need to contact us. Okay?
Yeah, so we can contribute significantly. Louis is still here. He's the PI lead of EUCAIM. Louis, can you tell us, this is EUCAIM obviously is building on all the other projects that were presented today. So what are the main differences? How do you see this relation of the EuCanImage and CHAIMELEON to the EUCAIM?
Well, thanks for that, Regina. Within the AI for health imaging, that was the seed of EUCAIM, and we were there. These 5 projects were at the end. The coordinators were deeply discussing for years and constructing the infrastructure. Each one of the projects has a different scenarios, approaches, and solutions to data sharing, data transferring, distributed repositories, links to the hospitals, data holders. So I do foresee that we do have like all the spectrum of possibilities, how data, hospitals, institutions, and partners can be linked within the EUCAIM. And we are discussing, all of us, which is within the flexibility of EUCAIM. We are trying to do the best to link researchers, institutions, and data and tools, which are the individual troubles, and options, and also contracts and agreements that we have to do. So my message is that EUCAIM is designed to be extremely flexible. We are really willing to have an infrastructure that will help researchers, and if some project partner or institution is not willing to transfer data, then we discuss on sharing. If there is a low level of digital maturity, we do share the standards and how can we help to define their data warehouse. We really want to be an accelerator of the research. And having said that, there is no single solution to all of us. Solutions are, as medicine, personalized.
Yeah. Well, thank you. It's very, very clear. I really like the fact that these data sharing help us to get a different mindset, because in the past, before the digital period, we were so only working in silos and protecting the research data, right? But now it's a completely transparent data sharing. So yeah, it's a very good news. Can one of the experts, maybe Karim, can you take this question on harmonization? What is the best way to approach data harmonization?
Yeah, well, that's a difficult question. I don't know if there's a best way. I can tell you that in EuCanImage, the way we did it is simply by doing an inventory in the different clinical sites, carefully selecting a reasonable set of non-imaging variables that to include in our studies. So at the beginning we had a lot of variables, and then we reduced them. And I guess just to be sure, data harmonization, you mean the non-imaging data imaging, right? Or the imaging?
Yeah, the imaging, but also the link towards other biological
No.
data.
Oh, yeah, the patient information, things like that. So for the imaging, we don't necessarily harmonize. There are ways to harmonize the appearance of the images. So that's an option. There are techniques to harmonize the distribution of the image data. That can be done, and it can help build AI tools that generalize across different data sets, different sites. For the non-imaging data, it's very difficult. I mean, the way we did in EuCanImage is we used existing data models. So there are data models out there that you can use. So you select a number of variables, and then you use the existing data models in a way that you get a data set that is as standardized as possible, so other researchers can reuse them. So we describe the data in we transform our data set to FIRE. So that's the data model we used in our project. And I think in other projects, they use also FIRE or OMOP. So it depends on the project, but that's the idea. It's really to use an existing data model. It's not trivial. It requires a bit of work, but it's useful because then you have a data set that becomes interoperable, so to say.
Oh, perfect. A question to Maciej. You showed us very interesting mammography models. Can you for example, the source code, is that publicly available? That is one of the questions. Can this software be used for further research?
So from this specific paper, I think we reposited it, and it's mentioned with the link in the paper. The same with the rest of the research. We usually tend to publish not only the paper with the results, but also the source code as per requirements of the European Commission. So we try to always provide it.
Yeah, very transparent. So Anna, one question from a radiologist who says, starting experience in AI, is that okay to contribute, or should we, as radiologists, know a little bit more about the AI field and AI expertise? So very nice that people are really enthusiastic.
Yeah, for sure. No? I think this is quite broad question because also depends on the ambition of the radiologist in this sense using AI. However, today, there are many tools that are quite packed, I can say, that in order to be used in an agnostic way for getting all the technical details, using, for example, train an AI model, etc. So there are a lot of tools that is, in case the radiologist or the specialist interested in developing an AI model. However, if it's not developing the AI model, it's just using the AI model, for sure, it's not needed not to have technical knowledge on how the algorithm has been developed, has been trained. Of course, always is recommended not to have some knowledge in order to as well gain trustworthiness on how the AI works, and it's not just a magic wand. It's something behind that has been validated, designed, etc. So it's not needed, deep knowledge on the technical details. However, some knowledge on what AI can offer and which are the main goals that AI can solve. I think it's something that might be quite interesting for any specialist.
Well, thank you for this advice. And for that, I would like to refer the audience to the upcoming European Congress of Radiology 2025. It will, again, be a very interesting program. And those of you attending ECR know about all the sessions on AI, where you get at least the basic knowledge of what is AI and how does it work. And you will hear from all these experts here I'm sure that they will be giving you the information and the lectures. What I think it was very interesting, and time flies. I just want to ask every speaker to look into the future and give us an advice as audience. So 1-2 sentences, okay. The question is, what would be the future of these combined data sets, and what would be the clinical value for future research and even future practice? So can I just go from left to right? I see you on the screen first, Maciej.
So for me, the future AI is the predictive tools, to be true, because detection is there, segmentation, classification is there. I would like to know the future, if my patient will develop cancer, so we could concentrate really on prevention instead of treating, advanced stages of the disease.
Thank you, Maciej. I fully agree. Karim?
Yeah, for me, the future is really clinical translation. So this is all still at the research stage, a lot of it. And I hope that some of these tools, a lot of these tools, most of these tools will get to clinical practice. And for that, we need trustworthy AI, we need more validation. And this is where EUCAIM in particular, by linking all these data repositories and data sets, will play a major role.
Thank you, Karim. Very clear. Anna?
Yeah, my view is like similar, no? So as I see, the real value is the development of these AI tools for predictive and prognostic capabilities, no, in order to stratify patients and adequate the best treatment for the patient. And following up now with Karim's comment, being able to translate all these solutions into the clinical practice by building AI models that are more generalizable, not to other sites.
Thank you, Anna. Mario?
Yeah, thank you. I totally agree with my colleagues. I think the core message here is that this will allow unleashing the power of AI for better understanding the disease and also for prediction towards precision medicine.
Thank you, Mario. And last but not least, Luis.
Well, hi, Regina. My message will be the future. Well, we are the future, no? We are the present and we are the future. That means for me that we are, if we are physicians, engineers, the ones who are working today on these developments on precision medicine, trying to do the best, trying to improve how medicine is performed. We are the future. So don't be afraid of AI. Take advantage of what is being developed, be part of the team, be engaged on the platforms, take opportunities and be proactive. That will be my message if you want to be part of the future.
That is a very, very good conclusion. So I really learned a lot from all of you, all the speakers. I really would like to thank you for participating and sharing your outstanding achievements. So I'm very impressed, and I'm sure the audience are also. And audience, I hope you have enjoyed and learned also that your participation in these projects are very important. And as you've heard from Professor Maciej Bobowicz, you are the future. So please connect with them if you have questions or if you would like to contribute. You can see there the symbols. EuCanImage, EUCAIM, CHAIMELEON. So go to their websites and contact them, and I'm sure that we all will benefit from your contribution. And if you want to know more about the EIBIR, please contact us, contact me. And so far, I would like to thank you all again, and hopefully I will see you in 2025 in the next webinar. And I would, for now, wish you very happy holidays and a very good start of 2025. Bye-bye.
Thank you.
Bye.
Bye. Bye, everyone.
Thanks a lot.